All right, I see a lot of people already online, and we are 3 minutes past the start time, so I think it's about time that we get started. We have a little bit of housekeeping at the start anyway, so there is still time for more people to show up. So, welcome everybody to the 8th episode of the EcoGen webinar series. We are today talking about the role of microbiota in livestock production. And I'm Velma from the Norwegian University of Life Sciences, also working for HoloRuminant, one of the projects part of this EcoGen cluster. And I'm hosting today because I'm also a rumen microbiome researcher, so working on the livestock microbiomes area myself. So just a little bit of sort of general house rules for today before we get started with the science. First of all, please keep your microphone muted so we don't get any background noise. When we get to the questions and answers part, the discussion part after the talks, then of course you will be very welcome to unmute yourself and ask your questions yourself. You can also put your questions in the chat during the presentations if you have anything you'd like to ask, and we will come back to them when we start the discussion part. And then when we are in the discussion part, it's also a good way to raise your hand when you want to speak so we can see who has something to ask. And this whole session is being recorded. If you do not want your face on this recording, you can please keep your camera off so you will not be included in there. And if you have any technical problems, I will not be able to help you with that, but please refer to the chat where we have people who should be able to assist with those kinds of issues. So what we have on the agenda today, we are going through the introductions right here as I speak. And we have two speakers today with quite different microbiome-related topics. So first we have Wanxin Lai, who is also from HoloRuminant and NMBU, who is talking about kind of functional capacity in rumen ecosystems related to methane emissions. And then we have Urvish Trivedi from 3D Omics from Copenhagen University talking about spatial organization, microbial lifestyles, and Salmonella in an in vitro gut ecosystem. And afterwards, as I mentioned, we will then go on to have a discussion that will be with both speakers together once they are both done with their talks. But so we will then, without further ado, move on to our first speaker, Wanxin, who very recently defended her PhD. Congratulations to her again. And yeah, let's dive right into it, I would say. So Wanxin, the floor is yours. Please go ahead and share your slides.
Hi, my name is Wanxin. So this is part of my PhD projects. And yeah, so the title of my talk today is about functional capacity beats redundancy and why some human ecosystem emit less. So this work is basically a genome-centric view of how human microbial communities shape relevant phenotypes in livestock. So why is this work matter? That's the first question. So as many of you that here know that ruminant livestock like cattle, sheep are one of the largest source of methane on the planet. And methane is a potent greenhouse gas that has a much higher short-term warming effect than carbon dioxide. And crucially, almost none of that methane is made by the animal itself. It is made by the microbes fermenting feed inside the rumen. So this is basically really like three problems in one. And the first problem is the climate problems. And because of the climate problem, because of the warming. The second is the productivity problem because methane is lost as a loss of the feed energy that never reach the animals. And the third one is a microbial problems if methane is a microbial output. So the solution should start with the microbes. And in these studies, the animal was split into two groups using a threshold of 24 grams of methane per kilogram of dry metals intake. So high methane above that line, we call it HEC or LEC here. So keep that in mind because it runs through everything that follows. And this brings us to our research questions. How does the metabolic plasticity of the Rumen microbiome differ between the high and low emitters? The important phrase is that there is metabolic plasticity, the flexibility, or in other words, the capability of the microbial communities reside in the rumen. So instead of just asking which key species are present, this study asks what the whole community is metabolically capable of. So this is how the study was built. It is basically 6 steers, come from 2 different breeds, Angus and also Lewin, all fed with the exact same mixed forage and concentrate diet. So the diet is not a co-founder here. And then the rumen fluid was sampled at 5 time points over roughly 4 months. So this gives us 27 microbiome samples in total. Then we do a long and short read metagenomic assembles, the sequencing. So that gave us 700 max here. And I believe many of you will have experience with this kind of plot. And each rows here is a samples here. And what this bar shows that is the microbial compositions in the form of relative abundance, and then categorized into high and low emissions and 2 different breeds across different time points. And many of you will be very familiar with the fact that there is not distinctive patterns shows between high and low emissions because the microbial abundance data, they are pretty noisy. And then we only have 6 years here. So it is pretty difficult to actually spot some things that is persuasive enough. But this is pretty normal for us. And then each of the 700 genomes go through the pipeline. First, the genome is functionally annotated using DRAMS, which maps its genes onto the non-metabolic pathway from CAG and Metasys database. And then we use this software here called Distill R, so that all the pathway information is distilled down into a single number quantitatively, which we name it as the metabolic capability indexes. So this is the start of this genome-centric approach here. We measure the metabolic capability quantitatively using these MCI scores here. This MCI is simply the proportions of complete biosynthetics and degradative pathway of a genome encodes. So a high MCI score here means that a MAC is versatile, is a generalist, is capable of using a variety of substrates. And a low MCI score here, it means that that MAC is a niche specialist. So the MCI therefore gives us a strain level metabolic potential that the taxonomy and the bulk gene counts cannot provide by turning the microbiome into a quantitative functional trait. And what others usually do is that on the left panel here, you can see people will usually look at the alpha diversity, which tells you the within community diversity here. And you can look at the p-values, none of them are significant enough in terms of species richness, neutral alpha diversity, phylogenetic or functional perspective. And then what others will also do is look at the beta diversity here. So under the Hill framework, it extends the diversity measures by considering the species relative abundances. That's why on our right side here, we have 4 different t-SNE plot. And the first one here is the, is pure richness here. It simply means the diversity of the species, and the blue are those that are from the low emitters, and the red are the time series sample from the high emitters. And then neutral here is used Q equal to 1 is a Shannon entropy base. So it emphasizes the species proportional contributions, balancing the rare and abundant, rare and common species take that abundance into considerations, and you can see a very subtle differences in the centroid between the low and high emitters. And in terms of functionality, you also, and also the phylogeny, you still can see the differences are there, but they are so subtle. This is because we only have that little numbers of animals, and also the number of sample size is probably insufficient here. Next one. Hence, we propose using these methods, hierarchical modeling of species communities. So why are we using these methods? This is because this is because like the first of the reasons we don't have enough sample size. We only have 6 count. And HMSC, of course, it doesn't magically increase the sample size. However, it can correctly model the host level, random effects, and therefore it can take the hierarchical structures by accounting for the repeated measures, because we keep taking the same samples, the samples from the same host, and then we take the breed, we take the sampling time or potential covariates into considerations. So it can model the structures in a more accurate and honest way. And the second thing is that the final variance produced by these models, it allow us to see how much each of these covariates contribute to the final variance explained by these models. So here we can see that emission itself, it contributes 13% for the final variance explained using these models. And also for each of these parameters in these Bayesian models here, you can see that it actually doesn't produce a single estimate, but it gives you a range of plausible value of uncertainties. Because abundance data of microbiome, they are very stochastic and there's a lot of noise. And then there are phylogenetic relationship between the max. We have to take each of these into considerations. So there are so many uncertainty in there. We don't want just having a single value to represent the whole pictures. We want everything. And here, what I've been talking about, all these variants, these covariates, we are actually insert into these beta parameters here. All the covariates is fit into these parameters. And here we model the time point, the breed, the emission levels, high and low methane emissions, and as well as this split. This split actually, it talk about the protozoa-driven population clusterings. You can see there's 2 different clusters here. And if you're interested, we have published a paper in Nature that talk about it, but we're not covering it today. Lastly is the sequencing depth. This is another thing that you have to take into considerations. So this method is a more honest method that we think should be variables. It should be valued in more modern research, as well as the trait and environmental layer. These beta parameters, it can be further expand into this layer. It link the MAC by telling us the metabolic capability of the MAC. For example, what are the characteristics of this MAC that is found in the low emitters? We want to study them as a community. We don't want to study them as a single MAC, right? Since you're looking at the low and high emission categories. Okay, enough for the technical side. So for the result, so the most intuitive things that this method provide us is telling us the directional trends of this MAX in these 2 communities. It tells us whether this MAX is positive or negatively associated with the emissions. This is what beta diversity alone cannot tell you. So these models, it finds that under this phylogenetically tree, this low and high emitter can be separated into 2 clades. And clade 2s are those that are highly prevalent in high emitters, and clade 1 are those that are prevalent in low emitters. And you can see that clade 1 is dominated by Bacteroidota and also Actinobacteria. There are 220 MAC enriched in them. And for these that are enriched in high emitters, we found 191 of them. It shows a more diverse phyla diversities, and it has more Bacillota and Methanogens. here. And this confirms that the emission level is something can be spotted from the phylogenetic perspective that this model can provide to us in the very first glance. And then the second result, it moved from who is there to what they're capable of doing. So here I'm showing you another t-SNE plot. The first plot here is ordered by the 700 meg, it's the 700 meg ordered by the phylums. And then this is ordered by the metabolic capability, the scores that I told you just now, as well as what the output from HMSC tell us whether these clades, this is the clade that are enriched in the low emitters, and these are the clade that are enriched in the high emitters. And you can see that those that are associated with low emitters, it carry a higher metabolic capabilities. The score is high. It means that the MEC they are found in here, they are generalists. They are able to use a variety of resources, while those that are prevalent in the high emitters, they are more like a functional specialist. And quantitatively, the low emitters had 20.3% higher mean MCI than the high emitters, with a p-value that is pretty good. So there is a very robust differences. And let's not forget about these extremes of those that are prevalent in the high emitters. We can see that MECs like Patesi bacteria from the genus of Nanosynococcus, those have the lowest MCI. They are very prevalent. So this is pretty interesting because these MECs here, their functions are very special, and then they have a very low MCI, but then those that are thrive in the low emitters, they are basically functionally more neutral. So this is not simply more capacity everywhere, no matter in low or high emitters. It is basically the overall balance of the community that shift. You have to see them as a community and not simply just a MEC that is one or two MEC. And we also look at the functional redundancy perspective here. So here is the plot that shows the relationships between the functional redundancy of the MEC against the neutral alpha diversity here. So normally, more diversity, it means that the rumen or the gut microbiome has more functional redundancy. And then that means a lot of different microbes all doing the same job, which makes a community stable, but not very flexible. So the high emission cattle here follow the expected patterns. It has a higher curve than the low emitters. So it means that those that are found in the high emitters, they have higher diversity, higher redundancy, lots of overlapping rows. This is what we expected. But the low emitters is what brings us a surprise. So here I call it the redundancy paradox. This is pretty counterintuitive because those that are enriched in the low emitters, despite they are having lower diversity, they are having higher MCI scores, so they are generalists. They are capable of doing many things instead of less things. So it basically tells us that those that are thrive in the low emitters, they are able to do more with less or limited substrates. So it doesn't affect their performance. They're performing equally well, even though they have less members reside within. So they are basically all the multitasker genomes reside in the low emitters. This is the take-home message that the lean, high capacity, non-redundant community that is well suited to adapt to a variable feed supply. But what about the functional characteristics of this LEC and HEC? What makes this LEC so different from the HEC? So I'm giving you a giant heat map alert because this is what many people will do. So each of these row here, is the different functions from the SCAC and MetaSys database. So what people usually do is they will just trying to use their eyes to see whether there are any distinctive pattern, but then you're gonna catch some noise because sometimes there's just noise, it's not actual patterns. And now you're having 700 meg, you're not having 2 or 3 megs. So some people will also do like pathway enrichment analysis. But if you want to study all these megs as a community, you shouldn't, as what I have been repeating, you shouldn't see them as an individual, assume independence of the pathway enrichment. Then you're gonna miss out on a lot of co-founders or covariates variables, because all these members, all these megs, they covariate co-occur and respond together to a driver like methane. So a heat map or the simple overrepresented of pathway test, you couldn't see it. We use HMSC, we can see it. So how I see it, remember the trait, and we link the MAC to the trait, the layer, the four corner in the Bayesian formula just now. We can go back to that if you are interested about it. So to know what they are capable of doing, it reveals what they are capable of doing. So we have many pathways as what we have seen just now. So we fit them into the same models after 700 iterations. Then we can see that those that are prevalent in low emitters, they are basically able to degrade a broader range of carbohydrates. And critically, they channel the hydrogen through the succinate to propionate pathway. This is a hydrogen sink if you have been working in this field. So you know that it is succinate production, it diverts the hydrogen away from the methanogens. Meanwhile, for the high emission community, you run a lactate dominated strategies. So they are capable to produce lactate. The genomes, the MAX, they possess of extra copies of the lactate dehydrogenase enzyme, and we can see that there's a enriched lactate producers in these genomes as well, such as Streptococcus, Shapia, Candrelia, and in lack of those lactate utilizing microbes such as Megasphaera or Coprococcus. Hence, lactate accumulate. So it's not just about what I'm talking. I also have the metabolic profile that support the finding here. Although we only have 6 cow, right? We have sufficient powers to see there's a significant differences when it comes to lactate accumulations in the kettles. This is high emitters and this is low emitters. And this is the cake pathway. You can also see there are higher lactate accumulate and also more enzyme domains that is responsible for the lactate production as well. And then such differences you can see, although it is not significant difference here like the propionate and succinate, but then it tells us that succinate, the direction is that there are more succinate being used to produce propionate in the low emitters, but such differences you wouldn't see in the high emitters. So why it changed the methane emissions? Why does any of the changes, it can change the output? So it comes down to where the hydrogens goes, right? So methanogens, the archaea that actually make methane, do so by combining hydrogens with carbon dioxide. So any metabolic pathway that consumes hydrogen first, it acts as a brake on the methane. So in the low emitters, rumens, the hydrogen is captured. Active fiber breakdown feed the fermentations, and the succinate is produced as an alternative hydrogen sink. And the conversions of succinate to propionate consume hydrogen is automatically leave less hydrogen availabilities to the methanogens. And then when it comes to the high emitters, the hydrogen is left free. The fermentations lean on lactate. So very few microbes can take that lactate further. So it's basically a dead end for fermentation, and therefore the lactate accumulate. The hydrogen that pathway would have used stays available and the methanogen use it. So the same input, but different hydrogen phase that give us two different methane outcomes. So why is this work matter beyond the biologies? Because a genome-centric lens, it turns a vague statement like a less diverse microbiome into something concrete and more actionable using our genome-centric methods. We get a specific concrete microbial targets, the fiber degrading, the succinate producing taxa you might want to enrich, and also the low capability specialists that could serve as a marker of high emissions, as what we have seen just now from the high emitters. We also get pathway level levels, the succinate to propionate route and the hydrogen sink point to exactly where the fermentation could be steered. And rest assuring, this genome-based result mirror earlier genes and taxonomy-based findings so that using our work it at mechanistic depth rather than contradicting prior work. And because the MCI and HMSC framework is computational, so there can be scales and apply to a far larger cattle populations as the Rumen genomic keep growing. And of course, let's not forget about the limitations. We have only 6 count. Well, we are very honest here. And then correlation doesn't equal to causation, right? And also the functional trait are read from the genomes. So they describe the metabolic potential and not measure of the metabolic flux. And we also don't have the transcriptomic layer to confirm that the pathway is actually activated. And that's all from me. And thank you very much. And I hope you enjoyed the talk and learn something, take it back home. And yeah, thank you.
Thank you so much, Wanxin. You can stop sharing then. And
Okay.
since we don't have endless time, let's just get right onto it with the next talk from Urvish Trivedi, who is an assistant professor in microbiology at the University of Copenhagen. And we will hear a bit about Salmonella in an in vitro system, I understand. And again, please, if you have any questions, feel free to put them in the chat and we will come back to them once we get to the question and answer part.
Okay, from start. Okay, can everybody see the slides?
Looks perfect.
Yep, okay, fantastic. Okay, so today I'm going to talk to you about some of the work that we've done in a very large EU project called 3D Omics. So this is basically where a lot of the analysis that we've done in this project is stemming from. So 3D Omics is a Horizon 2020 Research Innovation Action Project. And what our goal was in that project was to focus on trying to reconstruct the three-dimensional landscape in the GI tract to study, host microbe interactions, but also microbe-microbe interactions and microbe-metabolite interactions. Obviously, this is not a small feat in the field. I mean, a lot of groups have wanted to do this. So it takes, a large number of people to do this. So the project itself consisted of 13 partners across 11 countries and also industrial partners. But the reason why we wanted to do this is because, I mean, a lot of the field, what the insights and everything that we get from it has been on the conventional omics layers. So usually, including myself, whenever I work with anything in terms of human microbiome research, we get a sample, we homogenize it, and then we study the system based on relative abundance profiles or absolute abundances if it's that layer is available. But one of the other things that is really appreciated in cancer biology is spatially referenced research. And I think this applies also to microbiome research because if you think about the intestinal ecosystem, it's not just a planktonic culture. It is very spatially structured. You have certain microbes that will situate in specific microhabitats. They will interact with their surrounding community members, and then they may never see the other co-abundant member that you see in your conventional metagenomics analysis. So 3D-omics was focusing on sort of this principle is can we actually resolve microbe and microbe feed and the microbe host interactions. Now this is easier said than done, but including a lot of the people that were involved in 3D-omics, some of us, we actually come from a microbiology background. So myself included, what I started doing a lot of the research with was actually looking at how bacteria interactions change when it comes to planktonic versus biofilm states. And these are the two opposing lifestyles that basically bacteria adopt ubiquitously. In one state, you have free-floating planktonic cells where there's rapid access to nutrients available. There's low cell-to-cell interactions because they're usually in like either small aggregates or they're single cells. Whereas in biofilm, you end up getting these really structured communities. You have a lot of cell-to-cell signaling. You also have distancing away from other members. You will preferentially sit next to a neighbor that you like. And this really has a huge effect on how the multi-species community functions as in general. So the one of the things that we focus on are multi-species biofilms in our line of work, because these biofilms, they're spatially structured, but they're also very ecologically complex. So you may have a monospecies biofilm. Let's say if Salmonella makes a biofilm, it'll be more resistant to antibiotics. But now if you have Salmonella that is interacting with the, host resident microbes, uh, how does that multi-species biofilm structure itself? Because it can have a huge effect on whether or not they compete or cooperate with each other, how they actually transfer mobile genetic elements from each other, and then also how they share nutrients. And this spatial heterogeneity, it's somewhat influenced by the successional patterns, the establishment, such as priority effect, which one comes in first, and also the evolution aspect, like which bacteria are going to be more closely related to each other or not. And this is really important in terms of infection biology when it comes to multi-species communities, because in your GI tract, you have a situation that is called colonization resistance versus polymicrobial synergy. Now, these two are two opposing phenotypes, but they're stemming from the same origin point. So let's say if you have an invading bacteria, Salmonella, coming in, if you have one community composition type, your community will either be able to outcompete the invading pathogen, and basically it'll utilize a lot of the nutrients that the pathogen relies on, such as maybe iron or mucin or whatnot. Whereas if you have another community composition type where let's say some species are missing or some of them are in higher relative abundance, you can have a synergistic effect where now instead of providing colonization resistance, it turns into synergy where your native microbiome is accelerating the infection phenotype. So this is what we actually, some of the principles that we wanted to study in 3D omics. So to do this, obviously, the project was just also a lot larger than just the spatial referencing aspect that I'm talking about. We also did a large scale animal trials in these experiments. But what I'm going to focus on today is the Salmonella trial. So the reason for this colonization resistance aspect, it's important, is because when you take probiotics or you're delivering probiotics into a system, you see that, okay, something is not colonizing, and then you assume that the probiotic has worked. But sometimes you may find that when you deliver the probiotic, you can't even detect a probiotic in the system after like 7 days, but you assume that it has somehow worked. So this is one of the reasons that we started a lot of these trials is because we wanted to study, okay, if you are delivering some of these synbiotics, prebiotics, probiotics, how does it change the landscape spatially, and how does that relate to infection status? So a lot of the animal trials that we conducted, they provided some of the baseline samples and foundational samples for doing a lot of the in vitro work, but also doing a lot of the spatial reconstruction data that we have from the GI tract. It goes without saying, I'm just the person that gets to talk about all this amazing research to you today. But much of this work was actually done by this fantastic postdoc, Andy Erga, and also his PI, Annalise. They're both from ETH Zurich, and they they have a really unique system set up at ETH Zurich that we collaborated together with them on to get this study out. So it was a two-part study where we've done everything in parallel and in concert, and luckily now this manuscript is actually already submitted and in revision. So what did we actually do? So we had a lot of these cecal samples and fecal samples throughout the entire GI tract from the animal trials that we did for the Salmonella infections. But rather than using a lot of animals to get the throughputness that we would need for the spatial analysis, Andy has this fantastic system that he works with. And this system, what it is the technological platform that we've used all throughout the rest of the study. So what you're looking at is a Polyferm S in vitro system. So the key innovation here is the use of porous polymer beads. And these are, imagine tiny gel spheres that are about 1 millimeter in diameter. And what we do is we actually take these beads and we inoculate the cecal slurry from the donor chickens that we have. These are fresh cecal slurries that we use. And then we mix it with the different trials that we've done here, which is with either the prebiotic, the synbiotic, probiotic, and the infection with Salmonella. And what the beads essentially end up doing is they'll immobilize the community on or inside them, but then the bacteria that colonizes the internal pore of the beads, that would function as your biofilm associated community. So you can think of this as sort of your gut mucosa equivalent, whereas the liquid fraction that you have in these fermentation reactors, you can think about this as your lumen equivalent because this is the planktonic fraction. Now, to simulate what happens in these chicken GI tracts, this entire fermentation reactor was run at 41 degrees Celsius and at a pH of 6, which would mimic the chicken cecum, at least from what we've measured. So whenever you have the community coming in, we first let it settle for about 2 to 3 days, meaning that we're not pumping fresh media in and causing the effluent to just go out because then you're having a lot of washout phase. Instead, what we do is we let it colonize, and then after that, we start doing the fermentation for 7 to 9 days continuously by pumping fresh media in and then collecting the waste out. Yeah. So, what did we actually do in this study design? So, the experiment was sort of structured like this. You have three individual donor chickens. So, COP basically means Copenhagen, Vienna means the ones from Vienna, and we had both of them. So, one of them is the Copenhagen one is 21-day old, and then the Vienna ones are 28 days old. And then each donor microbiota, they're run in parallel fermentation reactors, so all at the same time. They just receive different treatments. So the fermentation reaction 3 is our control, which is just the cecal microbiota that we got from the chicken and Salmonella. Nothing else in there, no treatment. FR2 is our reaction that we added inulin into, which is a prebiotic component at about 2 grams per liter. And then the FR3 is actually where we added the probiotics. Oh, actually, no, sorry. FR4 is the probiotics. And these are the 5 strains that you see listed on the right-hand side over here. Enterococcus, Pediococcus, Bifidobacterium, and so on. And then the FR1, it was actually our full synbiotic. Now, this is the inulin and all the 5 probiotic strains together competing against the Salmonella infection. And to sort of mimic what would happen physiologically, we had the ratio of the invading pathogen, Salmonella, at 10 to the third CFU per gram, compared to the Synbiotic or the probiotics, which were at 10 to the fifth, or yeah. So there's 100-fold difference between the two, which is sort of what you would expect during a pathogen invasion if this were to happen, because you're not, if you get a very high bacterial load, that would be unlikely. So the basically this was sort of our experimental setup, as you can see. Now, what did we do after we actually had this set up? So as you can see from this slide, Andy was very, very busy. So let me walk you through this complex slide. So from each bioreactor, we collected two main arms, which is the biofilm fraction and the planktonic fraction. Now, the biofilm fraction, which is the beads, they actually had three sub-arms. So what we did was we pooled some of the beads for colony counts, which was just CFU per gram that we measured. We also did 16S sequencing. We also did qPCR to measure Salmonella absolute counts. And then to not have just a community scale profile of all the pooled beads together, we also wanted to analyze individual beads, because here each bead would serve as sort of a macro scale habitat where there's micro scale habitats within them, which I'll show you later. So we also took 10 individual beads to quantify them with the species-specific PCR, so we know what communities are on each single bead. We also took about 15 to 20 beads at 72 hours to do confocal microscopy and also biofilm matrix staining. And then we also took the endpoint measurements as well. The reason that we did 72 and also the endpoint is because we were worried that we don't want to be measuring a concentration or abundance profile of anything that is stemming from a dead community. So we wanted sort of to mimic also like the exponential phase that we would see within the reactions. And then from the planktonic fraction, we collected the supernatant, which we did metabolite profiling on 16S as well, and qPCR. This combination was sort of split as like a bulk community analysis and also like the single unit, which is the single beads here. So now that we've talked about the bulk analysis and the community analysis, what do actually these beads look like? So here you have a schematic that's showing one of these polyfermous beads. And what you're looking at is this is a 1 millimeter bead that has subhabitats within it. So the red dot, what we classify that is one microhabitat. And in this microhabitat, we have what you would call the live volume, which is going to be the live cells, the dead volume, which is more towards the inner layer, the union, which is the two combined, and then outside, which is sort of the biofilm matrix that's forming on top of around the beads. And then you can see on the chart on the right that when you measure these different sublayers, the median size is around 45 to 55 micrometer per dimension, and it's roughly around 150 microns across the longest axis that we measure on these beads. So these are sort of the scales that we're working with. So when we actually do the measurements here, oh, that is not going. There we go. Okay. So here you have measurements from 4 separate beads. So as you can see in panel C, where we did the live dead stain, each dot represents a microhabitat. So we've here calculated hundreds of different microhabitats within these single beads. And what we're getting is roughly around 75 to 89% of the bacterial volume in these microbeads is actually alive. And those are the green, live lines that you're looking at over here. Whereas the combined fraction is the blue one, but in terms of the dead volume that you have within these single beads, it's roughly around it's 11 to 24%. So you can see that majority of the microhabitat is actually live volume, but whereas where you go more towards in the center, you're getting more and more of these dead cells, which is what you would expect when there's nutrient or oxygen depletion happening. So these cells would first start off as dormant cells, but then as the aggregate size of the colonies and communities, they get bigger, the center ones, they don't have access to a lot of the nutrients and oxygen then. And then these beautiful images that you're seeing on the right is panel D. You're looking at the 10x magnification of the whole bead in one structure. And then in panel E, you can see here that at 63x magnification, you have a lot of these live green and dead cells that are inside over here in the center. So this is sort of like the ratio of what it looks like when we're looking at live versus dead fraction in these microhabitats itself. Yeah. And then there's also so now we have like the bead internal structure itself, but then there's also the community that's surrounding the bead, which is the attached biofilm layer. So this was also an important aspect because just like in the GI tract where you would have penetrating through the mucosal layer, you also have stuff that's on top of the mucosal layer and also surrounding the plant polysaccharides and everything. So here what we did was we used a matrix specific dye. We basically ended up staining the extracellular polymeric matrix here, which is the magenta color. And then alongside it, you have the fluorescent bacteria in green. So when we actually quantify this across the three different beads, you can see that the matrix volume is a lot larger than the actual cell bacterial cell volume that you have. So it's not really as if you're getting co-aggregates of bacteria that are just co-aggregating, but then they're actually also secreting a EPS matrix surrounding this bead panel. And then in panel G, this is again a 10x image where you can see that this magenta matrix right over here is surrounding all these cellular aggregates. And then in H, when you look at it, you can see that when you have this circular magenta blob over here, inside it, the cells that are there, they're the nested bacterial communities within this biofilm matrix itself. So this is really interesting for us because this model it is really robust because you can do hundreds of beads within one single tube reaction, and then within one bead you have hundreds of microhabitats. And this architecture that we study, it what we actually will see the next set of data is it really allows the medium and the metabolites to flow between the sub-communities and also during the continuous fermentation, which makes it a really dynamic system that you would expect to happen hopefully in vivo as well, right? So this was one major component of the study is to make a high throughput platform where we could study a lot of these spatial interactions. But the second part of the study was what actually happens when you have these competition dynamics with pathogen invasion and not. So the second part is, does the synbiotic treatment that we were originally looking at in these chickens, does it actually matter? And does it suppress Salmonella? And does it differ based on lifestyle state? So for that, let's look at the this figure here. You have the biofilm fraction. On the right, you have the planktonic fraction, which is the effluent. And each of the groups for the dots, they're actually from one donor microbiota, where blue is a control, red is the inulin treated, green is the probiotic, and purple is the synbiotic, both the prebiotic and probiotics combined. Now let's look at the planktonic fraction first. You can see that the Synbiotic, it inhibits Salmonella across all three of the donors, and the inulin and the probiotic alone, they also work, but generally less so. The Synbiotic is doing something that the individual two components cannot do by themselves. But if you look at the left panel, biofilm, the picture is completely different now. In the Synbiotic inulin, it looks like Salmonella is increasing in the biofilm. Um, and in the Vienna chickens, it looks like there's not really much of a difference. So remember, this is on bulk measure. This is what we've done by taking all the beads together, hundreds of beads together, homogenizing them, and then measuring on them. But what if we do at a single bead level? So instead of pooling the 15 to 20 beads that we did in the previous plot, here we've actually analyzed 10 individual beads per treatment and also measured by qPCR. So what you can see is in the Vienna chickens, the Synbiotic actually reduces the Salmonella, even in the biofilm fraction, by about 1.3 to 1.2 log cell copies per nanogram of DNA. And we were wondering why this is happening because likely, I mean, what is happening in the single environment is the spatial heterogeneity, right? Because in some beads, what might have happened in the previous plot is Salmonella was the first one to get in there, and then the other community compositions or the probiotics never had a chance to colonize. Whereas in some of the other beads, you have this priority effect happening where certain species are colonizing before Salmonella, and this is why you're getting these differences. So if this is really driven by spatial heterogeneity, we wanted to start diving into that aspect as well. So this is just looking at the Salmonella fraction based on the treatment. But when we also look at the metabolites, that also changed. So when we looked at the metabolites, we saw that when you have the Synbiotic and the inulin treatment, they produce higher metabolite concentrations of the three major short-chain fatty acids, which is acetate, propionate, and butyrate. They all go up. And in the Synbiotic, the inulin alone is actually higher than that as well. And then when you look at across the treatments, this is on panel B, you're just looking at a summary plot of this to look at across all the samples. But what we're actually really excited about here is because these, short chain fatty acids, they are known, particularly butyrate and acetate. They do have antimicrobial activity against Salmonella. They can also increase the epithelial barrier integrity of the gut. They lower the local pH, which can also disrupt the Salmonella membranes. And other studies have also showed that this can help reduce the Salmonella colonization. So it's not just the prebiotic inulin that's feeding the probiotics, it's actually remodeling the metabolic environment of not just the probiotic community, but also the cecal community in general itself. So to verify that, we get into the specific taxas that were driving these community shifts. And what you can see here is this is just a differential abundance analysis comparing the Synbiotic treated reactor to the control. And what we're seeing is on the left panel, the biofilm fraction, the taxa that are really enriched are the five probiotics itself, and then their log fold chain is about 5 to 15 higher. And then in the Vienna chickens, the 009, you can see that Salmonella is appearing on the left-hand side, meaning that it's getting depleted whenever this symbiotic treatment is actually being implemented. In the planktonic ones, the enrichment is a little bit different. It's producing, for some reason, it was producing the it was promoting the growth of some of the spore formers, but they're also butyrate producers, which are Eisenbergella, flavonorefractor, but these are not our probiotic community members. But either way, it is enriching some of these short chain fatty acid producers that was not achieved by a single treatment alone. But then now that we've shown this and we know that some community profiles are changing, and then also the probiotics and prebiotics are going up and down depending on the community member, we wanted to look at how they actually structure relative to each other. So you can see here where we have looked at the microhabitats within the bead itself, and this is just an example. You can see that the Salmonella clusters, it prefers to cluster away from the other community members, which are the beneficial ones such as Bifidobacterium and Lactobacilli and Enterococcus and whatnot. And then when we take each single microhabitat in panel C, if you look at it, so when some of these beneficial bacteria are present there, you don't see Salmonella popping up. And if you do see it popping up, it's in very low percentage of the population within that microhabitat. So it's this, co-exclusion profiles that are happening within these microhabitats. So now that we know that this is happening in vitro and we know some of these metabolite concentrations and everything, what we're wanting to do next is using the same animals that we have used for the cecal slurry, we're actually now wanting to go back and do this for all the intestinal sections. So we have a study right now where we've actually shown the 3D omics technology itself for generating microscale metagenomics data. But the following study, what we'll be focusing on is we have Salmonella infected versus non-Salmonella infected, and also the probiotic treated chicken samples that we're getting the intestinal tissue sections from, and we're doing cryosectioning, and then we're diving into some of the same analysis that I showed you over here right now. So stay tuned for that. Maybe for the next EcoGen talk, I'll give a talk on that project. So once again, this would not be possible with these fantastic people that actually put in a lot of the effort and the work into this project. And this was not just us. I see in the participant list, actually, there's many of the other 3D Omics members also in here. So a huge shout out to them. And this was a massive effort done by multiple institutes. And with that, I'm happy to take any questions. Yeah.
Excellent. Thank you so much, Urvish, and thank you both of you for very interesting talks. So we are a little bit getting close to our closing time already, so not so much time left for questions necessarily. Let me first just take a moment to mention to all of you that we do have the next EcoGen webinar, number 9, is already coming up on the 3rd of June, where we have quite different talks that are not at all to do with microbes, but we have some dairy cows and different pig breeds discussed, for example, in light of heat stress, climate change. And if you're interested in this one or anything else that we are doing, it's also very good to subscribe to the HoloRuminant newsletter, where we will mention upcoming interesting events to all of you. So I will leave this QR code up, and I think we should also get a link in the chat for those who are interested for further information. But yeah, so if anyone is happy to stay a little bit longer, we can now take some questions from the audience to our speakers as well. We had a few in the chat already and a few replies from Wanxin. So if anyone has additional questions that were not replied yet, for example, please put your hand up and we would be happy to hear to either of our speakers anything that you would like to ask. Alternatively, I don't see any hands yet. I can also sort of start things off with a very general question to both of you. So both your topics are quite sort of possibly a little bit esoteric to the lay audience, quite basic research. So when someone comes and asks from you, what is the practical application? How is this going to help us making our livestock production better or more sustainable or more in any way improved? What is your answer to this? What is the sort of downstream usefulness of this research? Maybe Urvish, you can go first.
Yeah, so I mean, so I think one of the things to consider is like whenever you have, infection happening with Salmonella, at least in mammals, in humans and mice, what happens is they actually use the Paris patches and M cells to transcytose through into the systemic fraction of the body. And now this is something that we really don't capture whenever we're doing like the bulk scale analysis. So if we were to deliver probiotics, prebiotics, maybe we have to somehow deploy them in like there's also this pH gradient right now, right? So there's more and more research into these sampling protocols that are pH dependent. So maybe somehow we can also lyophilize some of these probiotic strains and have them be dissolved in the right pH fraction in the GI tract and also the prebiotics as well, how to stabilize them. Because just just getting past the stomach seems to be the main priority right now, which understandably so. But I think then once it's in the GI tract, upper or lower GI tract, this is also where infection biology changes completely. And this is also dependent on the animals itself. So what might work in mammals is not going to work in maybe poultry systems. Yeah. So I think there is more and more need to generate spatially referenced data in gut microbiome research.
Excellent. All right. Wanxin, would you like to comment on sort of the practical implications of you?
So basically, my work is about two animals eating similar diet may process hydrogen and carbon very differently because of the microbial network organizations. So this could practically support like different feed strategies for different herds, diet formulation that favor succinate to propionate routing, maybe early detections of fermentative inefficiencies. I think that could be very useful for microbiome-informed animal breeding programs. For example, if certain microbiome states are consistently associated with low methane yield and better feed efficiency, this signature could eventually become biomarkers in the breeding programs. That's all for me.
All right, thank you. And this is a tiny bit touching maybe on the question also that you received, whether animal breed or physiology plays actually any role in these high and low emitting animals. Would you like to maybe comment a little bit on that also? You did write an extensive reply already, but...
Sorry, the first two questions?
Yeah, the first question that you got, so is breed or physiology important in general?
Yeah, they are all important because as we're looking at the microbiome perspective, it's not simply one meg driven everything, but they all work together. So there are different factors that can shape the final microbial metabolic configurations. So using this genome-centric approach, we can really see how much each of these covariate influence. So we know which one maybe we can prioritize more or pay more attention in the future. And for the next questions, yeah, so in sheep studies, right? At first when we see this result, we are pretty surprised as well because usually more lactate is associated with low methane emissions. But on the other hand, we also saw something that is pretty different is that we lack of lactate utilizer in those cows that have high methane emissions. We don't know why these microbes such as the megasphaera is not there. We don't know. We have no answer for that, but we can see how the hydrogen's alternative since changes there can change the entire pictures and also change our previous understandings on many things.
Right. So this was in reference to the second question, which was about some results of Wanxins that were kind of contradictory to some things that have been seen in sheep. So maybe also a difference in between species potentially.
I can see one of the question for me, if you are still there.
Very last minute question. If the person asking wants to stay online, you can still reply.
Okay, sure. I'll just give you a very quick answer for that. So in this study, I will not interpret the result being primarily driven by feed effect because the animal will maintain on the same mixed forage concentrate diet. So since all animals are on the same diet, I think the signal is less likely to be feed driven per se and more reflective of a host-associated microbiome ecosystem differences. So I would I would prefer to interpret it as a coupled host microbiome effect and not animal effect. It's nothing purely on each side. That's from me. Yeah.
Okay, great. One last bonus question to close things up.
Okay. Thank you. Yeah.